Hello, my name is Carlos Brian Francia, and welcome back to another lecture video. Today we're going to be learning about the basic anatomy of the human skin. This may look like a complex picture, but it's really not. We're going to start at the very top of this image, and we're going to be focusing on the epidermis. There's really three layers of the human skin. The first two layers are the epidermis and the dermis, and then the third layer is called the hypodermis, and that's where you see all the fat. Under the hypodermis, you have the fascia, which is the connective membrane, and then under that is the muscle, and then bones, and what have you. But right now we're going to be focusing on the epidermis, because the epidermis has different layers to it. And then after that, we're going to be talking about the dermis itself. When we look at the upper portion of the skin, we're looking at the epidermis, but the epidermis can be divided into 4 or 5 different layers. What do you mean by 4 or 5 different layers? Well, sometimes this layer, called the lucidum, is not present within thin skin. It's only present within thick skin. So if you see a stratum lucidum, you know that you're looking at thick skin. But where do we find thick skin? Thick skin is often seen within the palms and within the soles of the feet. And notice that these palms and these soles, they don't have hair on them. So typically when you see stratum lucidum, you shouldn't be seeing any hair follicles. But why do we call it a stratum lucidum? Well, lucid just means clear or transparent. And we'll talk about why in a few minutes, but really there's a special protein inside the stratum lucidum that gives it a transparent appearance under a histology slide. Now going back into the original epidermis, we have this little mnemonic and it says, "Come on, let's get sunburned." Well, each letter corresponds to the different layers of the skin. So for burned, we have the letter B, and B stands for the stratum basale. So the stratum basale is the basement, you could say the basement layer of the skin. That's where all the cells originate. So we have something called basal cells, and these basal cells are going to start making keratinocytes. Keratinocytes. Well, cytes just means cells, and these cells are producing something called keratin, and that is a fibrous protein. And this protein is very strong and tough, and it provides protection for the skin. In fact, your skin is made up of keratin. Keratinocytes also provide the waterproof effect for the skin. So if you have water here, the water is going to have a very difficult time trying to get inside the body, because these keratinocytes that are dead are providing the waterproof protection. Within the stratum basale, we have these little keratinocytes, and these keratinocytes are going to start dividing via mitosis. And they start dividing. When they divide, they're eventually going to go up, further up the layers. So they all travel up. And as they travel up, they eventually lose their nucleus, and they start producing some products. Like, for instance, we'll learn later what product this is, but eventually they start going into the upper portion of the epidermis, and that's when they flatten out and they become even more tough. They become dehydrated and flat. And we can see that there are different layers of keratinocytes here. So if it's layered, we call that stratified. And they look flat. When we call something flat, we call that squamous. And since they have keratin, we're going to call that keratinized. And therefore, the skin is made up of something called keratinized, stratified, squamous epithelium. And again, we want something to have a lot of layers to protect us from any trauma. Or if we rub our skin, we're not going to rub our skin cells off unless you have some sort of abnormality. That's a different disorder. but we want it to be keratinized so that it's waterproof and it is strong. So our skin is made up of keratinized stratified squamous epithelium. Also present within the stratum basale are something called melanocytes, and melanocytes are going to produce something called melanin, and melanin is just going to produce pigment. So the skin uses melanin as pigment. So we have different skin colors. We can be darker skinned or lighter skinned, and that is because of the melanin that we produce. But why do we want to produce melanin in the first place? There's something called ultraviolet radiation, and this is present within the sun. So we have the sun right here, and the sun's going to be a happy guy. You know, we have the sun right there, and then the sun is going to produce some rays, and some of these sun rays are going to go down into the skin and interact with the keratinocytes. However, too much ultraviolet radiation can damage the skin cells, it can damage the DNA, and it can damage folic acid. Folic acid is also a B vitamin, and specifically it is B vitamin 9, so B9. We need vitamin B9 to produce DNA. Ultraviolet radiation is going to damage DNA, and it's also going to damage folic acid. That's why we don't really want to be exposed to too much UV radiation, because if you don't have any B9, you cannot create any more DNA. Also, when you damage the DNA of the keratinocytes, they have a chance of rapidly dividing, and they don't stop dividing, so that can cause cancers, for instance, basal cell carcinoma or even squamous cell carcinoma. And that's why we produce melanin in the first place, because melanin is going to block the absorption of too much UV radiation. And when we talk about UV radiation, we have to talk about the three types of UV radiation. We have UVA, we have ultraviolet B, and then we have ultraviolet C. And again, this is produced by the sun, all three of them. A vast majority of the ultraviolet radiation is going to be blocked by the atmosphere, specifically the ozone layer. The most dangerous would have to be the ultraviolet type C, but that is blocked by the ozone layer for the most part. So we're really exposed to UVA and UVB. When you get sunburned, you are mainly exposed to UVB. However, you're really exposed to UVA when you're sun tanning. So sun tanning, let's actually do that in a different color, and let's do that in pink. So when we have sun tanning booths, that is going to be exposing the body to UVA. And if you use sun tanning booths chronically for years and years and years, you significantly increase the risk of developing skin cancers. So when you're studying for step 1, there are questions sometimes that say sun tanning booths are going to expose the body to what? Is to type UVA, and that increases the risk of having basal cell carcinoma and even squamous cell carcinoma. When you get sunburned, you're really exposing the body to UVB. So sunburns are going to expose the body to UVB. An easy way to remember this is that sun tanning booths, so we have sun tanning booths, are going to expose you to type A. So sun tanning is exposing the body to UVA type radiation, and that's not good. So I'm very adamant about people not using sun tanning booths because it increases your risk for skin cancer. We have to ask ourselves, how does melanin get inside the keratinocytes in the first place, and why does it occur anyways? Well, when the sun is producing its little UV radiation, the UV is going to be interacting with the keratinocytes. The keratinocytes are going to become irritated, and they start creating cytokines, which are little messenger cells, kind of like you're sending a little text message to the other cells, and it says, hey, I'm being exposed to UV radiation, I'm going to send this text message to the melanocytes so that they can increase the production of melanin. So keratinocytes are being exposed to UV radiation, they become irritated, they create more cytokinins, and these cytokinins are going to be interacting with the melanocytes. The melanocytes are going to start producing the pigment melanin, a little melanin right here, and then the melanin has to be transported to the keratinocytes. So the melanin are going to enter a taxi, and that's called a melanosome. So this is a melanosome right there, and you can see it over here. Now the melanosome is going to be filled with little melanin particles, and then is going to enter a keratinocyte. Inside the keratinocyte, we have to get rid of this melanosome. So now we're going to expose it to a lysosome, and the lysosome is going to start breaking down the melanosome using enzymes. And now the pigment is going to be dispersed in the keratinocytes. So here we have kind of like a darker color for the keratinocytes, and here we have a lighter color of the keratinocytes because not as much melanin is being produced. If we look on the left portion of the picture, we have some melanosomes right here, but then we also have some little melanin particles, and that's because some of the melanosomes have been popped from the lysosomes. So the little melanin pigments are just kind of hanging out inside the keratinocytes. And so to recap, when the skin is exposed to UV radiation, the keratinocytes become irritated, they release cytokinins. The cytokinins are going to be messengers for the melanocytes that increases the melanin production. And now the melanin is going to go into a melanosome pops using lysosomes, and it releases the melanin inside the keratinocytes. So the darker skin is a result of exposure to UV radiation. So if you have lighter skin and you go out in the sun, maybe you go to the beach for 5 hours and you don't wear any suntan lotion or sun lotion, then you're going to be a little darker when you come back home because you were sunburned, and the keratinocytes are trying to protect the skin by asking for more melanin production. And some people are going to try to increase their skin color by going to sun tanning booths or using suntan lotion, and that's not a really good idea because, again, you're kind of exposing the body to more UV radiation, and that increases the risk of DNA damaging and skin cancer formation over a chronic use period. I also want to mention that the human body, no matter what skin color we are, we produce the same amount of melanocytes. But you're probably wondering, well, why are some individuals a little bit darker than the other individuals? It really goes down to how our DNA expresses itself. So you have DNA is variable. So for this individual right here, that DNA is saying, hey, the melanocytes should produce a little bit more melanin. And for this individual here, the DNA is saying, hey, the melanocytes should be producing a little less melanin. We have the same amount of melanocytes, but the DNA tells us how much melanin we produce. And this is important because the more melanin we have, the harder it is to increase the vitamin D creation. So we have a decrease in vitamin D synthesis. So for instance, individuals who are a little bit darker may have a more difficult time creating vitamin D from the exposure of UV radiation. And if you have a decrease in vitamin D synthesis, then you have a decrease in calcium absorption. And therefore we should be a little bit more careful in our patients who are a bit darker because they might be vitamin D deficient, and therefore they might have a more difficult time absorbing calcium from their diet. And of course, I always misspell something. That's kind of like a little tradition that we have here, but that is melanocytes. We use tyrosine, which is an amino acid, to produce melanin. But how do we do that? Well, we're going to need an enzyme called tyrosinase. So we have tyrosinase, and ase just means enzyme. So tyrosinase is going to convert tyrosine into dopaquinone. And you don't have to memorize all the pathways for dopaquinone, but instead you have to know that we have two types of melanin. We have eumelanin and we have pheomelanin. Eumelanin is going to produce brown and black colors, and pheomelanin is going to produce yellow colors and red colors. So there's a whole spectrum. Depending on what the DNA codes for, we have different hair colors. We have black hair colors, brown hair colors, red hair colors, even blonde, and that's because of the different types of melanin that we produce. So for instance, I have black hair, so I'm going to be producing eumelanin, and that's going to code for black hair or for darker skin. So again, tyrosine is going to be interacting with tyrosinase, and tyrosinase is going to make dopaquinone. Dopaquinone could become eumelanin or pheomelanin. And then we have to ask ourselves, what happens if we got rid of tyrosinase? Well, if you got rid of tyrosinase, you don't have any dopaquinone. And if you don't have any dopaquinone, you don't have any melanin. And therefore, we can have a skin condition called albinism. Albinism. Notice this individual on the left-hand section. They are the same ethnicity as the other individual. However, the skin is not producing any melanin. There is no pigmentation. And this is called albinism. And that occurs because there is no tyrosinase. And again, tyrosinase is going to convert tyrosine into dopaquinone, and dopaquinone can become eumelanin or pheomelanin. If there is no tyrosinase, there is no production of dopaquinone, and therefore there is no melanin production. So that equals no melanin. And if we have no melanin, we have an increase of skin cancer risk, because melanin is going to be blocking the UV radiation. If we have no melanin, a lot of the UV radiation can enter the skin and it can damage the DNA and cause skin cancer. Also notice that this individual's hair is blonde. Well, melanin is going to be inserted inside the keratinocytes of the hair. So hair is actually made of dead keratinocytes. So we have keratinocytes. And these keratinocytes use melanin for the hair color. Since we have no melanin production because there is no tyrosinase enzymes, then the keratinocytes do not take in a color, and therefore they have a blonde looking color. And hair is pretty important for the body because it blocks a lot of the sun rays. So normally the sun would be right here, it'd be having a little sun rays right there, would go on to this person's scalp, and it would be blocked by the hair that has a lot of melanin in it. However, since this person has albinism, there is no melanin, and so when this person goes outside to play or maybe go to the beach or something, the sun rays are going to be kind of penetrating the hair, and the UV radiation is going to be damaging to the person's scalp, so that further increases a person's risk for skin cancer because there is no melanin production. And some ways that a person with albinism can live normally is that they have sun lotion and they protect themselves from the sun, or they wear hats, or they use umbrellas because they are very sensitive to the sun. It really hurts them easily. They have no protection against the UV radiation. This skin condition is called vitiligo, and vitiligo occurs because the immune system is attacking the melanocytes. So the immune system attacks melanocytes. This person can produce melanin, however, the body starts attacking the melanocytes at different portions of the body. So for instance, the body has not attacked the melanocytes in the cheek. However, in the left hand, we notice that the melanocytes have been attacked by the immune system. And if you attack the melanocytes, they can no longer produce melanin. These individuals who have vitiligo, they are also at an increased risk of developing skin cancer if they are exposed to too much UV radiation. Moving upwards towards the epidermis, we have something called the stratum spinosum. And the stratum spinosum, it kind of sounds like spiny or spines. Why do we say that? Well, these spines, you can see them right here. They're like little lines between the keratinocytes. But what are they? Well, really, those are the desmosomes, And whenever you have two cells, these cells are often connected to each other using something called an anchoring junction. So we have something called an anchoring junction. And that anchoring junction is called a desmosome. It's kind of like super glue for the cells. The more desmosomes you have, the stronger the connections the cells have to one another, and it is harder for them to be ripped apart. And these guys, it's easier if you kind of have a nice picture. So let's say that we have a keratinocyte right here, and we have another keratinocyte right there, and they're going to be little people. They're going to have arms, right? They have arms, and these arms are going to be connected to each other, and then they're going to start holding hands. And when they hold hands, they form something called a desmosome. So that is the desmosome right there. But what are the arms called? Well, the arms, they are called tonofilaments. These tonofilaments are going to be found throughout the keratinocytes, and the tonofilaments are going to connect to the desmosomes. And that's a really great picture or an example if you need that to study. So let's make our little tonofilaments right there. We're going to have the desmosomes right here, and then the tonofilaments are going to be found throughout the cell, and they're going to be connected right there to the desmosomes. This is the histology of the stratum spinosum, and notice that you have your little spindles right there, and these guys are the desmosomes. So that's when you're holding your friend's hand and you form a connection with them. That is the desmosome. You see that under the microscope slide. And then the desmosomes are connected to little filaments called the tonofilaments. And you have some right here too. So these are the arms of the keratinocytes. The arms are going to be holding hands with one another, and they hold hands, and when they hold hands, those are called the desmosomes. So when you're looking at a histological slide of the tissue, you'll notice that the stratum spinosum has a lot of these spindles, and those spindles are just the desmosomes. And the desmosomes are going to be leading into the tonofilaments of the keratinocytes. And you can kind of see some spindles right there, little connections, the tonofilaments right there. Really cool. If we go further up into the epidermis, you'll notice that there's a decrease in the desmosome connections. So in the beginning, at the stratum spinosum, there's a lot of desmosome connections right here, and right there too. But you'll notice that these connections are absent within the stratum granulosum and the stratum corneum. And that just kind of happens because if we had a lot of these connections to our neighbors, it would be more difficult for the skin to flake away. And so by having a decrease in desmosome connections, that allows the skin to flake off. And we want that to happen for the stratum corneum. Within the stratum spinosum, we have some special macrophages. We have something called a macrophage. And macrophages, they just eat the bacteria that enter the skin. And the name of these macrophages are called Langerhans cells. And here you'll notice that these little Langerhans are dyed with a special type of dye. And that just allows us to visualize it easier. So here is the Langerhans, and then it's going to have a long dendritic arm. So this long dendritic arm is going to be looking around for some bacteria. And let's say that it actually does find a bacteria. Let's say that it's right here. It's going to take that bacteria, it's going to gobble it up, and then is going to process that bacteria to present antigens. So Langerhans cells are antigen, they are antigen presenting cells. And that allows us to mount an immune response to any type of disturbance. We are looking at something called contact dermatitis. And contact dermatitis is usually associated with allergies or chemical exposure. But when we're talking about allergic contact dermatitis, that means that something touched your skin and you are allergic to it. and now the body's trying to mount an immune response to it. So let's say that somebody gives you a ring, and that ring is made of nickel. Whenever you put that ring on, the skin reacts to the nickel. And now the little Langerhans cells that you see here are going to get that nickel, and they're going to kind of make antigens for it, and now the body's trying to mount an immune response to the nickel. So here we have this irritation. We can see a clear definition right there around the circumference of the finger, and we can see the irritation of the skin. We also see some flaking of the keratinocytes within the epidermis. And mainly there's a lot of different immune processes happening here, but one of the immune cells inside the skin is called the Langerhans cells, and we can see them clearly because of this dye. And we can see that some of the Langerhans cells have these little dendritic arms, these little processes that are trying to take up the nickel and present them as antigens after processing. But how do we treat contact dermatitis? We can treat contact dermatitis by using a medication called clobetasol propionate, and this is something called a corticosteroid. And what this does is that it's going to decrease the immune response. So that allows the immune response to stop attacking the skin as much. And it's very strong. So whenever you use clobetasol propionate, you might have a burning sensation for a couple of minutes, and then that burning sensation goes away. And you typically want to do this for like maybe 4 days, and it should clear up that contact dermatitis. But it is a very strong corticosteroid, and you shouldn't use it too much because it does cause thinning of the skin if you use it over a chronic period of time. We finally get to the stratum granulosum. And granules, that just means little freckles. So these keratinocytes have little freckles in them, and that's why we call that the granular layer. So that is the stratum granulosum. But we have to ask ourselves, what is inside of these granules? It's actually something called keratohyalin. But we have to ask ourselves, okay, well, what does keratohyalin do? Keratohyalin further dehydrates the keratinocyte so that it can enter the stratum lucidum or the stratum corneum. Notice I said lucidum or the corneum. The lucidum is only present within the thick skin, such as the palms or the soles of the feet. And we have to ask, well, why do we call it lucidum? Why is it transparent? Well, it is transparent because it has a special type of protein. This protein is called elidin. Keratohyalin is going to convert into elidin, and then that elidin is going to contribute to the waterproofing of the keratinocytes. So that it contributes to the waterproofing of keratinocytes found within the stratum corneum. From the stratum lucidum into the stratum corneum, the elidin is going to convert into the protein keratin. So we have these keratinocytes producing keratin. To recap, we have the keratinocytes inside of the stratum granulosum that have tiny granules, and these granules contain keratohyalin. Keratohyalin is going to convert into elidin, and then this elidin is going to be present inside the stratum lucidum. If you do not have the stratum lucidum, you go from keratohyalin into the keratin protein. The stratum lucidum is the only layer that has elidin. If you do not have a stratum lucidum, you don't have elidin. And again, the stratum lucidum is only found in the thick skin, such as the palms or soles of the feet. We enter the stratum corneum, and the reason why we call it corneum is because that just means that it is hardened. So let's fully recap what we just learned over the course of 30 minutes. We have the mnemonic, come on, let's get sunburned. B stands for the stratum basale. The stratum basale has basal cells, and they are always mitotically dividing those keratinocytes. There's also the presence of melanocytes, and melanocytes produce melanin. Melanin goes inside the keratinocytes and give it a nice color. Then we go into the stratum spinosum. That's the S portion of sun, and the stratum spinosum has these little desmosomes all over the cells. And you also have some Langerhans cells. Let's just draw one right here. Let's say that this red blob is a Langerhans cell, and that allows the kind of immune response to happen. So maybe there's a bacteria. The bacteria is right here, is going to get nibbled on by the Langerhans, and then it's going to present the antigen to the T cells or B cells. After that, we enter the stratum granulosum. The stratum granulosum has these little granules. These granules are filled with something called keratohyalin. Keratohyalin can then convert itself into keratin, and you see that in the stratum corneum. However, if you have a stratum lucidum, let's say you take a tissue sample from the palm or from the sole of the foot, that keratohyalin will convert itself into ellatin, and that ellatin gives it a nice transparent looking color for the stratum lucidum. The keratohyalin further dehydrates the keratinocyte so that it can be flattened and stiff for the stratum corneum. And that's pretty much the basics of the layers of the skin. We just scratched the surface of the skin. After the epidermis, we have to enter the dermis itself, and the dermis has two layers. One of them is called the papillary layer, and that's more of the superficial layer right here. And then we have the reticular layer, and that's what you see here. It's very dense, and it has a lot of collagen. It has a lot of substance. One important thing that you have to realize is that the epidermis itself, has no vessels. So we can say that it is avascular. But you're probably wondering, well, if we have no vessels, then how does oxygen and nutrients get into the epidermis? Because if we didn't have any vessels, we would just have dead skin cells. And that's true. The dermis has vessels. So the dermis has vascularization. Here you have the lymph vessels, you have blood vessels, you have capillaries. And you also have nerves right there. And what happens here is that, let's say that we have some vessels right here. Let's say that that's a capillary. You're going to have oxygen and nutrients, and they're going to be leaving the capillaries. And when they leave, they're going to be diffusing into the epidermis. So the epidermis gets its nutrients, it gets the oxygen from diffusion. So we get O2 and maybe, let's say, nutrients via diffusion. When you're looking at a tissue sample, let's say that you're looking at a sample of skin under a microscope, I like to use landmarks for the dermis. So I can say, okay, well, this is where the dermis begins and this is where the epidermis ends. How can I do that? Well, I do that by using landmarks like this. So notice that we have some extensions of the dermis kind of poking at the epidermis. So this is what I'm focusing on. And that is called a dermal papilla. That is a dermal papilla. And we have an E right there, so the dermal papillae. And really, it looks like fingers. And I'll show you in a bit, but when you're looking at when you're studying histology and you're looking at tissue samples, you'll see these dermal papillae kind of extending into the epidermis, and that allows for greater connections between the epidermis and the dermis. The more dermal papillae you have, the stronger connections. So we have stronger connections. So we have an increase of connections. If you only have, hypothetically, one dermal papilla, then it would be very easy for the epidermis to be ripped apart from the dermis. Therefore, we're going to be using these dermal papillae as anchors. The more dermal papillae you have, the stronger the connections the epidermis has to the dermis, and the harder it is to rip the epidermis from the dermis. When we go further down into the reticular layer, you'll see that there's a lot of connecting tissues right there. They kind of look wavy. That's collagen, but there's also another protein that's very special, and that's called the elastin protein. And elastin sounds like elastic, and that gives the skin its elastic properties. However, in a clinical pearl, I guess something that you should look out for in a clinical setting, elastin has a high preference for bilirubin. So when bilirubin is being broken down, so we have heme breakdown, we have these byproducts of bilirubin. Some of them, bilirubin is yellow. So this breakdown of bilirubin releases yellow products, and that is being absorbed by elastin. So when we have jaundice, you'll notice that the skin is yellow because the elastin proteins have a high preference for the breakdown of bilirubin byproducts. You also have elastin within the eyes. So the white portion of the eyes, that's called a sclera, and it has a high density of elastin. So when we have jaundice, the byproducts of heme breakdown of the bilirubin is going to accumulate inside the elastin of the eyes and the elastin of the skin, and that's why we have yellowing of the eyes and the skin when we have jaundice. From here on out, we're going to be looking at histology slides, because when you're in medical school or in real life, you're not looking at very nice detailed diagrams, you're actually looking at tissue samples. And so here we can see this portion, we can see that we have the stratum corneum, and we're going to have the dermis right there. And if we go further into the tissue, we notice it kind of looks like a little net. And this net is actually the hypodermis. And it's very easy to tell because the hypodermis has a lot of adipose tissue. There are little cells inside the adipose tissue called adipocytes. So adipocytes just have a high fat content, and they look really circular. They have a really nice defined circular property. So in real life, here, these cells had a high fat content. And when you mix fat with water, which is present within the body, the fat forms little droplets that are perfectly circular. So in real life, when you're looking at histology, anything that has a perfect circular border was probably an adipocyte or adipocyte. Furthermore, we have the muscle. So we have this nice thin strand of muscle right there. You can see it's very nice and striated, which is classic presentation. So we can say that this right here is kind of like the fascia, the connective tissue of the body. So the muscle have this connective fascia, and that connects the muscle to the hypodermis, and then the hypodermis is going to be connected to the dermis, and the dermis is connected to the epidermis right there, which is what you see here. Let's zoom in and see what we can find in the epidermis. So if I look in the epidermis right there, we have all the layers of the skin. We even have the formation of the lucidum. So here we have the keratinocytes flaking away. They're dead. They're flat. This is stratified. You see the different layers. That is called stratification. And they're flat. That's squamous. And they have keratin. So we can say that the skin is made of keratinized, stratified squamous epithelia. And we can see that eventually these little nuclei are going to kind of float away. So initially the keratinocytes have a nucleus, but as they go closer and closer into the stratum corneum, the nucleus is lost, and you no longer have a nucleus within the keratinocytes. This nice gray presentation right here, that's the stratum lucidum. Sometimes in different slides, that stratum lucidum is going to be red. It depends on the dye and how the tissue was prepared. But in this sample, which is a sample from the University of Michigan Medical School, this is actually gray, and that is the stratum lucidum. It has a high content of ellatin, which is a transparent protein, and that allows the keratinocytes to be waterproof. Some tissue samples do not have a stratum lucidum. Instead, you're going to see the stratum corneum arising directly from the stratum granulosum. Speaking of the stratum granulosum, you can see it's a very nice, well-defined border. You can see it arises right here, and it's a very thin line of keratinocytes. And again, we have these little granules appearing because they contain something called keratohyalin, which becomes the ellatin you see here. If there is no stratum lucidum, then that keratohyalin just converts itself into keratin. If you do have the stratum lucidum, the keratohyalin converts itself into ellatin, and that ellatin converts itself into keratin. Moving on, you're going to see that we enter the stratum spinosum. So the stratum spinosum is directly under that stratum granulosum. And you can't really see it well because this is like the maximum zoomed in, but you can see that there's little lines between the keratinocytes, and these lines are desmosomes, and the more desmosomes you have between the cells, the stronger connections they have, and they're not easily ripped apart. Moving on from the stratum spinosum, you have the stratum basale, and the stratum basale has a high concentration of these basal cells, and these basal cells are going to divide and divide and divide via mitosis to create more keratinocytes. So here you have a high concentration of keratinocytes that arise from the basal cells. You can also see some keratinocytes accepting the melanin being produced by the melanocytes. And you can't really tell in this picture, but some of the melanocytes are a little grayed out. You can't really see it though. You might be thinking, oh, these are melanocytes. Really, there are keratinocytes, and these keratinocytes have melanin inside of them. Let's see if we can have a better picture right here. So you might notice that there's some brown pigmentation right there. That's a high concentration of melanin, and the melanin is going to be produced by the melanocytes. They travel inside the vesicles called melanosomes. The melanosomes are going to enter the keratinocytes and be kind of popped by the lysosomes. And when this happens, it releases the melanin inside of the keratinocytes. For people with vitiligo, the immune system is attacking the melanocytes, and therefore there is no pigmentation being produced or being given to those keratinocytes, and therefore it increases a person's risk of developing skin cancer if they are exposed to too much UV radiation. In contrast, a person would have albinism if there was no melanin production occurring, and that occurs because there is a defect in the tyrosinase enzyme. You need tyrosinase so that you can convert tyrosine into dopaquinone, and dopaquinone is then going to make eumelanin or pheomelanin. Eumelanin is just brown and black pigmentation, but pheomelanin goes from yellow to red pigmentation, and you would have also another increase in the risk of cancer formation due to overexposure of UV radiation. Moving on, we enter the dermis, and we can see the dermis protruding into the epidermis using the papillae. So this is a derma papillae. It looks like little fingers kind of pressing into the epidermis. You want to focus on the pink, not on the dark purple. Before, I used to focus on the dark purple and say, well, oh, the epidermis is going into the dermis. No, it's the other way around. The pink is going into the purple, and you can see that this is a clear defined border for the dermis. And when we enter the dermis, we enter the papillae layer first. After the papillae layer is traveled, we enter the reticular layer, and the reticular layer has a high concentration of collagen and also elastin. And elastin just allows the skin to be elastic, and it has a high concentration of connective tissues and proteins, and it's very beautiful. So here we have the nice dermis layer, the reticular layer, and it's all throughout here. You can see that, see? And then eventually you have more and more fatty content, and you go from the dermis into the hypodermis, or the subcutaneous layer of the skin, which has a high concentration of adipocytes or fat. Before I move on, I want to mention something. I want to mention that this is a dermopapillae, and it's a nice, almost circular looking formation. It kind of looks like a mountain right there. But what do we call this portion of the epidermis? What do we give it a name? We do, and that's called a rete ridge, R-E-T-E, ridge. And if you have any abnormalities in the rete ridge, you can have some diagnoses. For instance, if you have the elongation of this rete ridge, maybe it's further extending downwards over here, then we have the keratinocytes rapidly dividing. You have too much keratinocyte development, and that could lead to something called psoriasis. So what does that look like? This is psoriasis. So this is psoriasis. And specifically, this is actually plaque psoriasis. And we can see the elongation of the rete ridges. We can see that. And there is narrowing and elongation of the dermis, specifically the derma papillae. The derma papillae are narrow and they're longer because the rete ridges, which is what you see here for the epidermis, we'll color this in, that is called a rete ridge. So when you're looking at a tissue sample and you see elongated rete ridges, you'll know that, hey, the basal cell, the stratum basale, they keep producing a lot of keratinocytes, and when you have a lot of keratinocytes rapidly developing, they quickly rush into the stratum corneum. And when you rush the stratum corneum, you're going to have little nucleuses inside the keratinocytes. So normally, the skin takes about a month to develop, but in psoriasis, it takes about 7 days. So we're rapidly creating those keratinocytes and we're rushing them into the stratum corneum. Normally, the nucleuses will disappear. They're going to be shot out of the keratinocytes, and the keratinocytes are dead. However, in psoriasis, the nucleus is still present inside the keratinocytes because we're rapidly dividing. You can actually see a high concentration of keratinocytes within the stratum basale. So we have a high concentration of these keratinocytes, and that's very present within psoriasis. Also, as an aside, you can see some nice blood vessels right there. You can see some blood vessels, so you can notice that the dermal papillae have a high vascularization. So that's a very nice piece of information from this picture. Plaque psoriasis occurs because there is a rush in the development of the keratinocytes within the stratum basale. So we have a large deposit of keratinocytes within the stratum corneum, and you can see it's very flaky, it looks dry because there's a lot of accumulation of keratinocytes. We have the rapid development of those keratinocytes within a span of 7 days, and because it's rushed to such a degree, we still have the nucleus being present inside the keratinocytes, and it's very painful for the individual. So for instance, if this person kind of brushes upon a piece of furniture or maybe their clothes, this little area is going to kind of pop off. The little skin cells pop off and it exposes the skin. So now you have a painful lesion. You have a painful cut and laceration right there. So we have bleeding, irritation, just inflammation within the area. And since we have inflammation occurring, we can use something called clobetasol. So some individuals who have plaque psoriasis, they can use clobetasol cream, and they can kind of apply it to the affected area. And this allows the area to calm down and to stop producing so much keratinocytes. Again, you're still going to have these little flare-ups, and they occur when you notice these little papules. So these little papules are just small areas, and they start like that, and again it's going to grow and grow until it forms something larger right here, until it forms a patch of skin. So whenever somebody has psoriasis and they say, yeah, I'm expecting a new patch because I noticed this little dot. So this dot forms, and it keeps getting bigger and bigger until it forms a nice large patch. So again, that is from the overdevelopment of keratinocytes within the stratum corneum. Let's talk about sensation. So specifically, we have something called somatosensation, and that allows the skin to sense different items. So we can sense heat, cold, we can sense pressure, vibrations, even touch. But we have to talk about the different accessory organs that we have in the skin. So the first one is actually a Merkel disc. And a Merkel disc is the only sensory organ that is unencapsulated. So it has no capsule. So it's very, it just doesn't have a capsule. The other people have capsules. For instance, we have a Meissner capsule, which is called a corpuscle. And we also have the Krauss end bulb, which has a capsule as well. And we have the Pacinian capsule, also known as the Pacinian corpuscle. And again, we have Ruffini endings. But what do they do, and where are they found? A Merkel disc is found inside the epidermis, and it is associated with light touch, and we can say that it is slow adapting. So that is slow adapting. But what do I mean by that? what do I mean by slow adapting? And if there's slow adapting, there has to be something called fast adapting, but what does that mean? Slow adapting means that when you give it a stimulus, it will take that stimulus and it's going to recognize it. It's going to keep recognizing it for a long period of time. That is slow adapting. So let's say that we have a fly, and this fly is going to be on the skin. So we have a little fly right here, and we have another one right there, and maybe let's have an ant as well. So these guys are on the skin, and when they touch the skin, they sense, they send a stimulus to the Merkel disc, and that Merkel disc is recognizing the light touch. And because it is slow adapting, it's going to recognize those touches for a long period of time, and it sends the information to the central nervous system, and that relays the information to the brain, and it says, hey, there's an ant on the skin. I feel something crawling on my skin. It's very light. Let's move our body and smack the skin so we can kill that insect. So we send little signals to this Merkel disc, which has no capsule, so we can say it's unencapsulated, and it's going to be sending those light touch stimuli. It's slow adapting, so it's always going to recognize those signals, and then it sends the signal to the central nervous system, and the body can act upon it. But what about Meissner corpuscles? Meissner corpuscles also focus on light touch. However, they are quickly adapting, so that is a quickly adapting. But what does that mean? Well, these insects and flies, they are also sending out light touch signals. Initially, the Meissner corpuscle recognizes the light touch signals and it says, hey, there's an insect walking on my skin. But the longer that these insects send the light touch stimulus to the Meissner corpuscle, the less and less and less responsive we become. So eventually, when we have a prolonged stimulus, we just stop recognizing them. We just stop ignoring, we start ignoring the stimulus because we already had such a prolonged effect on the Meissner corpuscle. So that is the difference between quickly adapting and slow adapting. Slow adapting means that we can recognize those stimulus for a long period of time. Quickly adapting means that the stimulus over a short period of time is going to be ignored. So the more stimulus we have for a longer period of time, the more and more and more we ignore it. The Meissner corpuscles also respond to low frequency vibrations. What does that mean? Well, it means that, let's say that a car is blasting music with a nice subwoofer, and you hear that nice bass, and you can feel the vibrations in the ground, and when the car rides right past you, you can feel the vibrations inside your body. Well, that is caused by the low frequency vibrations, and the Meissner corpuscles are able to detect this low frequency vibration. So when we think about Meissner corpuscles, we think that they are encapsulated, they respond to light touch, they are quickly adapting, and they also recognize low frequency vibrations such as a car blasting music and it shakes your entire body. What about Ruffini endings? Ruffini endings also have a capsule, so they are encapsulated, and they respond to warmth. And if they respond to warmth, then they're also going to be slow adapting. Why is that? Well, we want to be slow adapting because we want to recognize when something is hot, and then we want to continue to recognize that it's hot. So for instance, if I'm making food and I put my hand on the stove, my body uses the Ruffini endings to say, hey, that stove is hot, I'm going to damage my hand if I keep touching that stove. So I'm going to send that message to the central nervous system and move my hand away. I am slowly adapting. However, if I'm quickly adapting, I put my hand on the stove, I recognize that it's hot, and then in 2 seconds, that sensation of hot or warmth goes away. And that's bad because if I keep putting my hand on that hot stove and I don't recognize that it's hot, or I don't respond to it, my hand is going to be burned and severely damaged. So we want something to be slowly adapting when it comes to warmth or when it comes to temperature. Ruffini endings can also be used to do a proprioception. What is proprioception? Proprioception just means that the body realizes where the limbs are at. let's say that you're exercising. When you're exercising, the Ruffini endings are sensing the movement of the muscles. They sense the movement of the joints within the knees and the elbows. And it sends that information to the brain. And the brain realizes, hey, my left arm is behind my neck right now. Or my left arm is behind my left knee right now. That is proprioception. It just allows the body to keep track of where the limbs are and what we're doing. So the Ruffini ending is used to also realize the proprioception and warmth information. And Ruffini endings are found within the dermis. Krauss end bulbs are found within the dermis. And here, the Krauss end bulb is only used for thermoreception. That is thermoreception. And specifically, it senses the cold. Cold. So if I were to hold an ice cube, that ice cube is going to be being sensed by the Krauss end bulb, and the Krauss end bulb also has a capsule on it. So the Krauss end bulb is only used for thermoreception, and it senses the cold. Finally, we have something called a Pacinian corpuscle, and this is found deep inside the dermis, and you can also find them inside organs. So for instance, inside the pancreas, or the breast, or even around the bones. And what they do is that they sense pressure, but not prolonged pressure. This is actually quickly adapting. So for instance, if you were to put your hand on your thigh and quickly press down and then press up, you sense that pressure, and that pressure is being sensed by the Pacinian corpuscle. It also responds to high frequency vibrations. So for instance, we have this individual using a jackhammer, and this jackhammer quickly vibrates, so he can feel those vibrations inside his organs and around his bones. So when he stops using the jackhammer, his body still feels like it's shaking, but then it quickly dissipates, it quickly goes away. And when you're looking at tissue samples, the Pacinian corpuscle looks like an onion. Kind of like when you cut an onion in half, it looks the exact same way for Pacinian corpuscles. What you're seeing here is the Meissner corpuscle. So here is the stratum corneum, the stratum granulosum. Notice the absence of the stratum lucidum. And then we have the stratum spinosum, and right next to it is the stratum basale. Under the stratum basale, towards the portion of the upper dermis, is the Meissner corpuscle. And this Meissner corpuscle has a high density of nerve endings. And you see a high density of Meissner corpuscles within the fingertips and within the soles of the feet. So for instance, if you're walking, you want to be sensing everything that you're touching. If you're touching something with your fingertips, you want to be able to sense it. So we have a high concentration of Meissner corpuscles inside the fingertips and within the soles of the feet. And here we have another nice picture of the Meissner corpuscle. Notice that it kind of looks like it has little layers inside of the corpuscle. And also notice how close it is to the dermis layer. This is actually in the papillary layer of the dermis. It's right next to it. It kind of looks like a little jelly bean inside of the papillary layer of the dermis. Here we have an example of the Pacinian corpuscle, which has a capsule, and therefore it is encapsulated, and it looks like onion slices. So we have different layers of the Pacinian corpuscle. And when you see here, around it, these little dots, they're actually fibroblasts. So the layers are made of collagen, and the inner circle of the Pacinian corpuscle is myelinated. That means it has a lot of nerve endings that have fat surrounding the nerves. So those are the myelin sheaths created by the Schwann cells. And since we have a high concentration of myelination, we have a high concentration of nerves. So these areas are rich in nerve endings, and therefore Pacinian corpuscles respond to high frequency vibration and pressure. And they're found within the hypodermis. Well, not in the hypodermis, but deep inside the dermis, next to the hypodermis. Moving on, we are now discussing sweat glands and sebaceous glands. Within the skin, we have different types of glands, but we're going to be focusing on sudoriferous glands. And in Spanish, sudor means sweat. So that's what we mean by sudor. Sudoriferous just means sweat glands. And sudoriferous glands are going to be a subtype. So they're going to be using the merocrine secretion method. But what does merocrine mean? Well, here's what I mean by that. There are 3 types of secretion methods. The first type is going to be the merocrine method. And what happens here is that the product is going to be created inside the cell and then is going to exit the cell via exocytosis. And then it's going to leave via a tunnel. And this tunnel is called a duct. And here, let's say that we're making sweat. The sweat molecules, or I guess the waste byproducts and electrolytes, that's part of sweat, they exit the gland via the ducts, and they exit the body from pores of the skin. So that is a merocrine method. And what uses the merocrine method are the sudoriferous glands. But what are the sudoriferous glands? We have 2 types, and they are the eccrine glands, and then we also have the apocrine glands. And both of them are going to make sweat, and that's why they're called sudoriferous glands. The other type of secretory method is called the apocrine method. And what happens here is that the upper portion of the cell starts to pinch off. So there are really 2 sections of the cell. There's the basal section, and then you have the inner section right here, and that is called the apical region. So it's kind of like the inner portion of the cell. And so whenever the product is being created by the cell, it goes into the apical region, it becomes pinched, and then it leaves the cell. It leaves the cell again through ducts. But where do you see that happening? You see this happening in mammary glands. So when a person is nursing a baby, the mammary glands are going to do apical secretion, and they're going to pinch off little globules of milk into the baby's mouth. So that is the apical or the apocrine method of secretion. A long time ago, people used to think that the apocrine glands, the sweat glands, secreted using the apocrine method. That's why they have the same name. However, that's not true. The apocrine glands actually secrete using the merocrine method. And when we secrete the sweat of the apocrine glands, bacteria are going to feed on the waste products, and they produce an odor. So that's why you have kind of like an odor to these apocrine glands. And where you find these apocrine glands is actually in the genitals, and within the axillaries. So for instance, within the armpits. So we would attribute odor to those regions of the body. And that's really because the byproducts that we release is being fed on by the bacteria. And finally, we have another method, and that's called the holocrine method. What happens here is that the cell or the gland starts to disintegrate. It starts to break down, and when it starts breaking down, it releases its product into the duct. So a good example of this would have to be sebaceous glands. And sebaceous glands produce something called sebum. And sebum, that's just a glycolipid that coats the hair and also coats the skin. It allows the skin to be further waterproofed, and it also gives waterproof properties to the hair. So usually whenever you have sebaceous glands, they're right next to the hair follicle inside the skin. Here we have the sweat glands, and we also have the sebaceous gland. The eccrine sweat gland and the apocrine sweat gland, they are considered sudoriferous glands, and they secrete via the merocrine method. For the sebaceous gland, that is going to be the holocrine method. And I want you to look at the anatomy really quickly. Notice that we have a duct connecting the sebaceous gland to the hair follicle. We also have a duct for the apocrine gland connecting to the hair follicle. So imagine if this is the armpit, for instance. The byproducts of the sweat is going to be dumped into the hair follicle and is going to leave right here. So that's what happens when we are sweating. In the case for sweat glands, the sweat that they produce is actually hypotonic. That means there's more water than solutes. But what are the solutes? The solutes can be IgA. This is an antibody, and it helps eliminate bacteria from the upper portion of the skin. So let's say that we have some bacteria right here, and they try to enter the body. The IgA antibodies are gonna surround the bacteria, and they prevent it from moving any further. So that's what the IgA antibodies do. Another solute that we make are electrolytes. So for instance, when we're exercising really intensely, we lose a lot of sodium, and the sodium is being transferred out of the body via the sweat. We also produce something called dermicidin. So we have dermicidin. So dermi kind of looks like dermis, or the skin, and then cide just means to kill. So this is antimicrobial. And that prevents bacteria from setting up little towns and colonies on the upper skin. So the dermicidin is going to kill a lot of the bacteria that kind of sit on top of the skin. And in the case of the sebaceous gland is going to make something called sebum, and sebum is like a little oil that coats the upper portion of the skin, and it allows the skin to be waterproof. So it further waterproofs the skin. What you're looking at right here are the eccrine sweat glands. Now, the eccrine sweat glands, they kind of look like little ovals, but they're small. And let's just put eccrine. And you'll notice something, that there are some blue strips surrounding the outside of the eccrine sweat gland. What is that? Well, what it is it's called a myoepithelium. So really, it's epithelial cells that have the properties of muscle. So it is muscle-like epithelium. And what it does is that it allows the epithelial cells to contract. And when they contract, they're going to squeeze. And when they squeeze out the product. They squeeze out the sweat. And that sweat goes into a into a duct. And that duct can go out of a pore. So these eccrine sweat glands have a layer of myoepithelium on the outside. So it would be right here. And that squeezes the sweat out of the eccrine sweat glands, and it squeezes the product into a duct, which goes into a pore of the skin. On the inside, what letter B is pointing to, those are the cuboidal epithelium. So we can say that the eccrine sweat glands are made of cuboidal epithelium, and they have an outer layer of myoepithelium, which are just epithelial cells that have muscle-like properties that allow the eccrine sweat glands to contract and squeeze out their product into a duct, which leads into a pore on the surface of the skin. This is the apocrine sweat gland, and the apocrine sweat gland again is going to be in the genitals and also in the axillaries. You'll notice that the apocrine sweat gland is a lot larger than the eccrine sweat gland, and you can see that this space right here, this space is called a lumen. That is called the lumen, and it's the inner space of the gland. And we can also see that some of the cells look like they're being pinched off, and you may even see some cells kind of floating around. Because we saw this, scientists thought that the apocrine sweat glands released via the apocrine method, but in reality they release via the merocrine method. When we produce sweat, that sweat is going to be dumped into the hair follicles within the genitals and within the axillaries. And the apocrine glands also have this thin layer of myoepithelium surrounding the apocrine glands. So here we have it. And all that does is that it's going to squeeze the apocrine glands, and when you squeeze them, they release their product into the duct. So yes, apocrine glands and eccrine sweat glands have a thin layer of myoepithelium. And then the inner layer is just simple cuboidal epithelium. What you're looking at is the tissue of breast. So this right here, all of what you see is breast tissue, and what I've just highlighted are the mammary glands. So this is a mammary gland, and as you know, these glands secrete using the apocrine method. The apocrine method. This is a gland right here, and what you're seeing here is that this is producing some milk. So milk is going to be kind of pinched off and is going to be released into the duct. Now, surrounding this gland is a thin strand of myoepithelium. So you can see that these glands all have something in common, and that is the myoepithelium. When a person is breastfeeding their baby, the milk globules are going to be kind of forming on the apical region of this portion, and the milk globules are going to eventually go into the lumen, that is the inner portion of this gland, and then it's going to go into the duct, and the duct is going to lead into the baby's mouth. And that's how a person breastfeeds their baby. So we can see that we use the apocrine secretion method, and then we have the myoepithelium in green squeezing the duct, or squeezing the gland, so that the product can go into the duct and feed the baby. What we're looking at here is actually a hair follicle, and surrounding the hair follicle are glands that produce sebum, and these glands are called sebaceous glands. Sebaceous glands. Sebaceous glands are going to produce their sebum, and they excrete the sebum via the holocrine method. So this is a holocrine method. And sebaceous glands are always going to be dumping their sebum into the hair follicle. So now we're going to be kind of secreting the sebum around the hair follicle, and it leads into the outer portion of the skin, and that sebum is going to coat the outer portion of the skin as well. So sometimes when you run your fingers through your hair, you'll notice that your fingers are kind of greasy. That's because of the sebum. You'll also notice that if you touch your nose, your fingers are also greasy. That's because of the sebum. And sebaceous glands are partially controlled by hormones. So they're also hormonally controlled. So when you were like 14 and you were going through puberty, you may have noticed that your face was a lot oilier, and you may have had acne. But when you're a kid, your face is not as greasy. That's because your hormones haven't really kicked in yet. So the sebaceous glands are really active after you hit puberty. You don't really have the activation of sebaceous glands as a child. And this is the hair as it forms inside. The way the hair forms is that it forms from the inner hair bulb. So inside the dermis, there's something called a hair bulb. And this hair bulb acts very similarly to the creation of the epidermis. So you know in the epidermis there's something called the stratum basale, and the stratum basale creates those keratinocytes. Well, we're also using keratinocytes inside of the hair bulb. So here we have the stratum basale of the hair bulb, and then it's going to be producing these keratinocytes. And these keratinocytes are going to start kind of forming until it forms a hair follicle. So you can say that hair is actually dead keratinocytes. And you're probably wondering, well, if it has keratinocytes, there's probably melanocytes. And that's true. along this region right here, you also have melanocytes, and melanocytes are going to start producing melanin, and the melanin enters the keratinocytes of the hair bulb. So as the keratinocytes are being formed for the hair, they're also taking in some of that melanin, some of that pigmentation, and that allows the hair to take on different colors. So for me, I have eumelanin, and eumelanin is going to be dark, maybe a black color. So now we have this black color forming right here. And what that does is that it allows the UV radiation of the sun to be blocked from the scalp. You don't want a lot of UV radiation on the scalp. That's a very sensitive area. And so we have a high concentration of hair so that we can block those UV radiations. So what we learned here is that hair is made from dead keratinocytes, and those keratinocytes are being created by the stratum basale. And again, they're going to divide via mitosis and eventually exits the scalp and shows as hair. And that hair is going to block those UV radiations and protects us from the sun. Here we have the hair bulb. So this entire region is called the hair bulb. And if we go inside the hair bulb, we have this section right there, and that is called the hair root. The hair root is where all of the keratinocytes are being developed. And inside of this hair root, we have kind of like this area right there. And this whole area is called the hair matrix. This is where all the magic happens. This is where the keratinocytes absorb the melanin and they start kind of stacking on each other. So we have the hair matrix. And what makes up the hair matrix is something called the medulla. The medulla, that's just the middle of it. And then we're going to have the cortex surrounding the medulla. Again, we have another cortex right there. So the cortex surrounds the medulla, and it goes and it continues upwards towards the hair follicle. And outside surrounding this, right here in this region, that's something called the hair cuticle. So that is the cuticle. And you should probably do that in a different color. Is this better? The cuticle. And that just surrounds the cortex, or I guess the hair matrix. And the cuticle is very firm and it's very rigid, and it gives the shape of the hair follicle. So when we have curly hair or straight hair, that is really determined by the shape of the cuticle. And inside here is actually something called the dermal papillae. Dermal papillae. And what this allows us to do is that it gives nutrients to this hair. So for instance, we have little capillaries right there, and then we have tiny venules. So oxygen can go inside and feed the stratum basale, and then it can take in waste products from the stratum basale and take it into the circulatory system. There's also nerves right here. So there are nerves right there. So inside the hair plexus, and the hair plexus is right there, is what we're looking at. We have vessels. We also have nerves. But notice that there are no lymphatic vessels for the hair. So the hair doesn't have any lymphatic vessels. So no lymph vessels. So the dermal papillae kind of goes inside the hair bulb, and with it is a region inside called the hair plexus. Plexus just means collection. But what is the collection? It is actually of vessels and nerves that give oxygen and nutrients and information for the hair. When you age, the amount of melanin production decreases, and therefore there's not as much melanin inside the hair. And that leads to the graying of the hair, or even the hair becoming white. With people with albinism, there is no production of melanin because there is no production of tyrosinase. There's a disorder of the tyrosinase enzyme, and because of that, there is no melanin inside the hair, and there is no adequate UV protection of the sun for the hair. So that increases the risk of skin cancer for those individuals. And we can actually see the hair plexus right there. This is the hair plexus, and we notice that there are some capillaries and some tiny venules and some nerves attached to this hair bulb. And we should also realize that there's something called an erector pili. This is an erector pili. And what it does is that it senses stimuli and is going to pull on this hair follicle, or I guess this hair follicle, and it's going to make the hair stand up. So instead of looking like this, the hair is going to start looking like that. And you might realize that this is goosebumps. And what that does is that it's going to kind of trap a layer of air. And that increases insulation, which increases warmth. So maybe you're in an area where it's really cold. Let's say that you're visiting Alaska. Well, you're going to have goosebumps because the body's saying, hey, we're cold, but if I pull the hair using my erector pili, I can trap some air right here. And air is a really good insulator, and it keeps heat trapped right here. So here's the red is the heat, and the air is the blue. So we trapped a thin layer of air, and we trapped a thin layer of heat, and we can use that as warmth. And that's pretty cool. So now you should be able to explain in detail everything that you see here and more. So I hope that you understand the basic concepts of dermatology, and now you can use that foundational knowledge when you study dermatology even more. So thank you so much for spending your time with me, and thank you so much for being awesome and cool. I love you so much, and thank you for spending your time with me. It was awesome teaching you. All right, take care. I love you, and have a great day.